Hello, students, and we are back with another chapter. Today we are starting with chapter 13. This one's a little more confusing again. This is talking about gene regulation, so we are going to try and clarify some of the more confusing points. Keeping in mind that we've been learning about genetics and how genes work and why genes are important, chromosomes and things like that. Now we got to understand that genes don't need to stay on all the time. You don't need all of your stuff working all of the time. You don't need all of your enzymes working all the time because it's not necessary and it's expensive. Like, energy is the resource that your body uses, and your body is constantly wanting to conserve energy. So you really don't want to spend energy if you don't have it, if you don't have to. It's kind of like money, and really is comparable to money. You want to save as much energy as possible. You want to save as much money as possible. You want to spend money if you don't have any extra to spend. You want to save as much as you can. And same is true for energy. You want to save as much energy as possible. So turn off the money and the energy if you don't have any, if you don't need it. So that's basically what this is talking about, is how genes are regulated, turning them off, turning them on as necessary, and also the causes of some mutations. So prokaryotic regulation, eukaryotic regulation, and also gene mutations. Gene mutations are pretty easy to understand. Those aren't too complicated, but the prokaryotic and eukaryotic regulations are a little bit more complicated. This is another one of those chapters that it might be helpful to go to YouTube and watch some examples. Before this, I was just kind of refreshing my own memory on some of these terms, like operons, for instance. And the Amoeba Sisters have a really good video on YouTube that is very helpful explaining some of these topics. So they do a good job of like adding some humor and explaining things in a way that's easy to understand, with some fun videos. So definitely check those out to help clarify some of these things. But basically, for prokaryotic regulation, bacteria basically do not require the same enzymes all the time. Enzymes are basically produced as needed. So more complicated organisms like us, we often have a lot of those enzymes present in our body more frequently, and we just turn those enzymes on and off as necessary. But prokaryotic, they're much smaller, so they just don't need the enzyme. So they will build the enzyme when necessary. Whereas with us, we keep the enzyme and we just turn it on and off whenever we need it. But Francis Jacob and Jacques Monod, they propose that there is this operon model to explain regulation of genes expression in prokaryotes. An operon is a group of structural and regulatory genes that function as a single unit. So this means that this is a series of genes that they all kind of work together to perform a specific goal. In this case, turning something on or turning something off. So these regulatory genes tell the body that, okay, we need to get this process started, or okay, we need to stop this process. And again, the purpose of that is we might not need this particular resource all the time, or we might just need it occasionally. So a regulatory gene is normally located outside the operon, and repressor proteins control whether the operon is active or not. When we're looking at these sequences, there's a premotor, and the premotor is usually kind of the first step, and the DNA sequence where the RNA polymerase first attaches. Remember that the DNA is the code that all of this information comes from. The DNA is the code that holds all of the genetic information, but also all the proteins and all the enzymes and everything comes from this DNA sequence. And so it's kind of like the go-to blueprint for all of this information. And the premotor has that, is found on the DNA sequence as well. And so the RNA polymerase, remember, anything ending in ASE is a type of enzyme. RNA is going to go to that, and it goes to this premotor area, and it's going to find that and kind of attach to this area. And it's really just a short segment of DNA telling it, okay, we're going to kind of start here for this thing to work. The operator is next, and the DNA sequence where active repressors bind. So this is where the binding is taking place, and it's another kind of short segment of DNA. So just like a short chunk of DNA, just like the promoter is another short segment of DNA. Then you've got your structural genes, and these are one of several genes coding for enzymes of a metabolic pathway. And they're kind of transcribed simultaneously, kind of as a block, kind of as these chunks again. And these are longer segments of DNA, so they are longer, but they are kind of in these long, chunky pieces. So the regulator codes for a repressor, and there's different kinds of them. So this example is looking at the TRP operon. Tryptophan is an amino acid, and this is looking at specifically this one. So different kinds of operons operate for different functions of genes. So for tryptophan, and I'm sure you might have heard of tryptophan before. Tryptophan is found in like turkey meat, and specifically you hear this around Thanksgiving time. Tryptophan makes you tired, and that's true. And it is found in turkey, which is also true. But if tryptophan is absent, the repressor is unable to attach to the operator, and the expression is normally going to be on. So if tryptophan is absent, the expression is normally on. If tryptophan is present, it blocks the synthesis of the enemies, and then the expression is therefore going to be off. So that's kind of how tryptophan works in this case. If tryptophan is absent, the amino acid is absent, the repressor is unable to attach to the operator, and the expression is normally on. The enzyme for the synthesis is produced. If tryptophan is present, then this blocks the synthesis because the enzyme is already there, and the expression is therefore off. So this just kind of shows how it works. Here we've got our regulator gene, and here we've got our active repressor. This is the promoter area and our operator. And so we've got our active repressor that's going to come here and bind to it. When the repressor binds to the operator, transcription is prevented. Now remember, transcription and translation are part of the protein synthesis process. So if transcription is prevented, that means no protein is going to be made now. If transcription is prevented, we cannot make a protein. So it's preventing a protein from being made. It's preventing this structural genes, this DNA sequence, from being read. So no protein is going to be made as long as this repressor is here. It's not allowing protein to be made. Now this we get if the tryptophan is absent. So now we remove this. We can actually make this mRNA strand from this segment of DNA, and now we can make the necessary proteins. But we add this on, and now we've blocked it. So we put this tryptophan on there, and that tryptophan is present. The presence of tryptophan prevents the production of enzymes used to synthesize it. So when it's present, now we've blocked it and we can't produce any more. But when it's absent, it's no longer being blocked, and we can make that. It's no longer being repressed, it's no longer being blocked, which means this opens it up, and that means that it can be created and protein synthesis can take place. When tryptophan is present, it's blocking it, and protein synthesis cannot take place. Now, in this case, this is lactose operon, which is a lac operon. And lactose, a sugar that can be used for food, is absent. The expression is normally off. If lactose is present, then it combines with the repressor and renders it unable to bind to the operator. The 3 enzymes necessary for lactose's catabolism are produced. So in this case, it's a little bit different. In this case, we see that without the lactose, it's binding, and that's kind of opposite of what we saw in the previous one. Okay, so without it, it's binding, and no lactose can be produced. This time with it, with the lactose present, it's removed, and now it's opening up that ability to make that enzyme. So this one, it's kind of flip-flop from what we saw from the TRP operon. Okay, so that's a little bit different. So it really depends on what kind of gene we're looking at, what kind of expression we need. With the lac operon, if the lactose is not there, then we don't need that gene to be expressed. So we can repress it with this repressor here. But if the lactose is there, then it binds, and that means we do need that gene to be expressed. We need that enzyme to be made, and so now it binds to that, it removes that, and now we can make that gene. Whereas if we go back to the other one, if this tryptophan is present, that means we don't need it. So the two are opposite of each other, if that makes sense. There's other further control of the lac operon. There's other things that control it. So this just means that sometimes gene expression, there's more than one way to control it. There isn't just one size fits all. Sometimes there's more than one way that can control it and keep it, either stop it or start it from being expressed. So E. coli preferentially breaks down glucose. The lac operon is maximally activated only in the absence of glucose. When glucose is absent, we have the cyclic AMP or CAMP accumulation. CAMP binds to the cap, and this is where it gets a little confusing. Actually, that video, I can't add it into my video because of copyright laws, but the Amoeba Sisters on YouTube explains the lac operon pretty well. When CAP is bound, RNA polymerase binds better to the promoter. The structural gene of the lac operon are expressed more efficiently. So it's just further control. Sometimes more in the area activates it better and helps it to produce things more efficiently. So that's basically all this is saying. All right, so when glucose is present, there is little CAMP in the cell, and kappa is inactive, and the lac operon is not expressed maximally. So here we can see inactive CAP. Okay, here's the promoter, here's the operator, it's the binding site. But we can see it gets more complicated. Promoter cells, here's a promoter, there's the operator. We can see a more complicated going on here, but we're trying to open up this site so that we can actually make the proteins necessary and the enzymes necessary. All right, so there's more that goes on with eukaryotic regulation. So there are 5 primary levels of control. There's nuclear levels, cytoplasmic levels. So with nuclear levels, there's chromatic structure, transcriptional control, post-transcriptional control, translational control, and post-translational control. Those are in the cytoplasmic levels. So transcription and translation are 2 parts of the protein synthesis. So transcription takes place within the nucleus, translation takes place within the cytoplasm, and that's kind of why they're broken up into nuclear and cytoplasmic levels. So the nuclear level takes place within the, his transcription takes place within the nuclear level, and translation is the second step of RNA synthesis, protein synthesis, and that takes place within the cytoplasmic level. So, eukaryotic regulation can take place anywhere within that. And we have the chromatin structure, the transcriptional control, post-transcriptional control, translational, and post-translational. So this just kind of shows a picture of all of the different places that we can monitor the regulation of genes. It can be in many different steps within this process in eukaryotic cells. So first of all, the chromatin structure. So the chromatin structure itself is how the chromatin is made. The chromatin is made by being wrapped up in nice, neat little bundles. First of all, you've got a histone. A histone is a protein, and you have a nucleosome, which is the histone is the little protein, and then a nucleosome is the DNA wrapped twice around that little histone. So if you have the DNA is wound around groups of 8 molecules of histones, so if you have 8 of those, like little beads on a string, this is what this is talking about. So 1, 2, 3, 4, 5, 6, 7, 8. So this is one little grouping that it's referring to back here. So DNA is wound around groups of 8 molecules of histone proteins. So then we've got these little groups of 8, and here's our histone, and 2 loops of DNA around a histone. This is a nucleosome, okay? So each one of these is a little nucleosome, and the purple is the histone, and that's just kind of how DNA is organized. And we've got DNA kind of bundled together like this, and this is all going back to just, it's how DNA is organized, because if you have this just long, stringy DNA, it can get really tangled up and really hard to transport around. It's nice and neat and bundled up, kind of like if you go to a craft store and you were to buy yarn, it's easier to buy yarn that is wrapped up in a nice, neat bundle. If you were to try and buy yarn that was unraveled, the yarn would be like trailing through the store, getting tangled up. And then if you tried to undo the yarn to like crochet or knit something, I mean, it would be in knots everywhere and you just get frustrated with it, trying to untangle it. Same idea with DNA. The DNA is so long and stringy, you need it wrapped up in a nice, neat little bundle so that way when you need to unwind it for something or you need to transport it, it's ready to go. And that's what these nucleosomes are doing. Wrapping it up nice and neat around 2 little histone proteins, and then you've got this nice, neat little bundle ready to go. Euchromatin is loosely coiled DNA, transcriptionally active. So that means that it's coiled so loosely that you can have transcription take place. And transcription is when we can get DNA unzipped and get an mRNA strand in to get the information from the DNA strand. That's transcription, is getting the DNA information onto an mRNA strand. That's transcription. So this euchromatin is when we can still have that process take place. Heterochromatin is more tightly packaged, and this is transcriptionally inactive. This is not active enough that it's too tightly packaged. We can't get the information. Now, sometimes this is a problem when it is too tightly packaged. We can't control it. So, females, for instance, have 2 X chromosomes, but only one is active. Excuse me. Only one is active, and the other X chromosome is not active. It's too tightly packaged, and that we consider a bar body. So one of the X chromosomes is the active one, and one of them is inactive and is considered the bar body because it is inactive. It's a heterochromatin and tightly packaged and does not go through the transcription process. So here we can kind of see how that works in cats. We have our inactive X chromosome that is much more tightly compacted. It still has the information on it, but it's just so compacted together and tightly woven that it's not able to unwind enough to go through the chromosome process of making proteins for the body. But we have a much longer and bigger chromosome that can be used for making proteins. So a histone modification may be linked to epigenetic inheritance. Epigenetic is when you have multiple characteristics coding for a gene in which variations in the pattern of inheritance are not due to changes in sequence of the DNA nucleotide. Epigenetic inheritance refers to inheritance patterns not dependent on the genes themselves. So the genes themselves may not necessarily be represented in the genetic inheritance. Explains unusual inheritance patterns. So it may also play a role in growth, aging, and also in cancer. Now you could have transcriptional control. So during transcription, there are things in transcription that can control how a cell is monitored. Remember, we're still looking at how cells are monitored and regulated. Proteins that help regulate transcription by assisting the binding of the RNA polymerase to the promoter. So a transcriptional activator might be necessary, and this binds to the DNA protein, and it binds to the enhancer DNA. So we've got a transcriptional activator, and we've got an enhancer DNA, and all of these are going to play into making a copy of the DNA. The enhancer, but it's most likely to have to be activated before they will bind to the DNA itself. So here we can see kind of how it works. We've got our enhancer over here in this long DNA. Remember, DNA is very long. Promoter, and then the gene. And then we've got our transcription activator with our mediator proteins. And then the transcription factor complex, our RNA polymerase. All of this is combining together to try and make sure that our transcription process can proceed correctly. Now, if it does proceed correctly, we can still control the process by post-transcriptional control. This is a series of, and I know we mentioned this before, introns and exons, and splicing these. The introns are actually the ones that are moved away, and the exons are the ones that stay and get shoved together and kind of meshed together while the introns are removed. And so this is another kind of way we can control areas that might be allowed to stay within that mRNA or that might be removed. May also control the speed of RNA transport from the nucleus to the cytoplasm. So this helps with, can we make the mRNA cell or mRNA strand quickly versus slowly, and if we can make this process speed up or slow down. It will affect the number of transcripts arriving at the rough ER, and therefore the amount of genes produced per time. So we can make this process faster or slower and make more or less genes depending on how fast we can move this post-transcriptional control. So here we can see two different proteins, protein product 1 and protein product 2, and we can look at how this is being spliced and sliced and put together. And we've got our introns and our exons and building them and combining them, and sometimes we can do it faster and sometimes we can do it slower. The non-coding transcription DNA is used to form small RNAs, or sRNA molecules, involved in gene regulation and function at multiple levels. So this is another way that genes can be regulated. This is the RA interface, and this is referred to as RNA interface, and small interfering RNAs. So this kind of shows that process. We've got a DNA here, and then a dsRNA after alters the DNA compaction, can kind of break it down into small RNAs, and those small RNA pieces can be inserted in, and that can also help with the movement out of the nucleus. We could have translational controls. Now remember, after transcription comes translation, and this is the actual combining of the protein using the messenger RNA that we got from the DNA and taking it out of the nucleus and into the cytoplasm. Translational control is determining the degree to which the mRNA is translated into a protein product. Features of the mRNA affect whether translation occurs and how long the mRNA remains active. So we can determine how long the mRNA is going to stay alive within the cytoplasm, and whether it's going to break apart and go back to its base pieces once it reaches the cytoplasm. The length of the poly A tail on the 3' end will help to determine that. If the poly A tail is longer on the 3' end, then that means it will stay stable in the cytoplasm longer. If it's a short poly A tail, then that means it will break down sooner. And we have post-translational control, affects the activity of the protein product, activation and degradation. And proteases are enzymes that break down proteins, and by doing so they help regulate gene expression. These are confined to proteases or lysosomes to protect the cell. Just some other examples. A lot of times ATP is necessary for the combination of these things and adding molecules to them, so we can see ATP being used here. And the caps that are put on to help them make them more stable. All right, now moving on quickly to gene mutations. Gene mutations are processes that can happen during many points within the transcription, translation, and replication processes. Sometimes they can have no effect on the protein activity, or they can have complete inactivation of the protein itself, and the protein just doesn't work and doesn't finish developing. Germline mutations occur in the sex cell, so sometimes these mutations can occur in the male and female gametes, or they can occur in the body cells as well. So these mutations can be random, they can be genetically inherited, they can be something that happens during the development of the sex cells of the body. So spontaneous mutations are chemical changes in the DNA that lead to misbearing during replication. So this may be that there was an error in proofreading the strands and the strands just didn't match up correctly. Generally, these do get corrected. Overall mutations are 1 in 1 billion, so that's actually really good that our body only makes one mistake in 1 billion nucleotide pairs that are replicated. And a lot of times those one mistakes aren't even that serious or can be fixed. So the fact that our body is that precise is pretty impressive. Induced mutations are caused by mutagens, radiation, or organic things such as food and tobacco smoke, carcinogens, which are cancer-causing, again, such as like sunlight that damages our skin, or tobacco that makes it, gives us lung cancer. There are many things within the environment that are unhealthy for us. So those are induced mutagens, but there can be spontaneous mutations as well. Here's just the Ames test for mutagenicity. Suspected chemical mutagen, which is added to bacterial strain, and then you put them on a petri dish that lacks histidines, and you observe whether they grow or not if incubated overnight. And if they do grow, that means there was a mutation, and if they do not grow, there was no mutation. There's also something called a point mutation. One type of point mutation is a base substitution. A lot of times this is going to happen, could happen in replication, transcription, or translation, where a nitrogen base gets switched, and if a nitrogen base gets switched, it could change one codon to a different codon, and the effects vary. There could be no effect. It could be something like a silent mutation where no effect is detected, or it could be unaffected overall. There could be a frame shift mutation where 1 or 2 nucleotides are either inserted or deleted from the DNA, and sometimes it can be very serious, such as you can see here, the cat ate the rat, because these are all grouped in threes, and groups of threes are called codons. So the cat ate the rat, but here you can see the atta to her at. So just by deleting one thing, so we deleted the C here, but it threw everything else off. In this case, the insertion, the cat ate the rat, we inserted a C here, and by inserting an extra C, we threw everything off as well. So that's how serious these things can get. This is showing what the normal, original DNA strand look like, and a base substitution would look like here, an addition, and then also a deletion. So lots of different things going on. A point mutation in hemoglobin, we just have one mutation that's changing, but that one mutation also changes the thing it's bonded to, and that can result in an overall different type of mutation, in this case, sickle cell anemia. If a faulty enzyme is inserted into a metabolic pathway, a person may be able to convert one molecule to another with serious consequences. Phenylketonuria, or PKU, is an example of how this works, and it can eventually cause intellectual disability. Androgen insensitivity, and that causes female instead of male genitalia to form. Mutations can cause cancers. Oncogenes, which stimulate cell division, and if they get mutated, then they can stimulate excessive mutations where they don't, the body can't control them. And mutagens and oncogenes, and tumor suppressor genes, if those get malfunctioned, then tumors can grow without the ability to repress themselves. It stimulates the cell cycle to continue uncontrollably, and this can often lead to tumor formation, not always cancerous, but a lot of times cancerous. Okay, there we are, finished with chapter 13. Hopefully you find that helpful, and we'll go on to the next chapter next time. Bye, guys.